Assalamualaikum and good day to everyone. This is our fifth lecture on virology, and we will look into vaccination and its significance toward minimizing the spread of various viral disease. This topic belongs to the second CLO of this course, which is to evaluate the types of viral diseases, vaccination concepts, and development of virus vaccines. What is vaccine? The word vaccine is actually derived from Vaccinia virus of the pox virus family, which was used as the first ever vaccine by Sir Edward Jenner to prevent smallpox infection. The vaccine is supposed to be able to stimulate immune reaction in the host body but doesn't cause any infection or disease. This is called active immunization. If you look at this figure, vaccination is categorized as an active method to provide artificially acquired immunity by manipulating our specific immune system to produce antibodies, thus preventing future disease. So, the main difference between natural infection and vaccination is that the person doesn't become ill when immune system is responding to the virus, because the virus is either weak, inactive, or incomplete. Look at this timeline. In fact, the concept of vaccination was first introduced by Chinese several hundred years before Edward Jenner's first experiment, and it is called variolation, which is to prevent smallpox as well. In this practice, they take the fluid from pox blisters on skin, dry it for a few days and grind to make them like powder, and then they will inhale the powder. If lucky enough, the inhaled powder will contain non-viable smallpox virus, which stimulate immune response but doesn't cause disease. In unfortunate cases, the virus still viable and infect the person. After smallpox, one by one vaccines that we know today being introduced since 1800s until the latest HPV vaccination in 2010. And we are yet to see the introduction of first dengue vaccine, which is set in its final trial phase and to be released. As you see, there are several types of vaccines available, and we will go one by one. Live vaccine is a vaccine that uses living virus. The only live vaccine ever existed was smallpox vaccine, which used cowpox instead of smallpox virus. Cowpox only causes mild symptoms to human, and antibody produced against cowpox is able to recognize and bind to the smallpox virus as well. Second, one of the most common type is attenuated vaccine. In this type, the virus is being weakened by treating them with chemical up to the level where they lost the capacity to cause disease. Since they are not dead, they are able to enter the host cell and thus are able to stimulate both cell-mediated immunity and humoral immunity as well. The third type, inactivated vaccines. This type of vaccine use that virus. Their structure remain intact, so they are still able to trigger antibody response, but doesn't trigger cell-mediated immunity as they are not able to enter the host cell anymore. The advantage of this vaccine type is their safety. It can safely be administered to immunocompromised individuals without chances of causing any disease. Toxoid vaccine is mainly used to neutralize toxin produced by some bacteria, such as tetanus and diphtheria toxin. So far, toxoid is only used against bacteria. Polysaccharide and polypeptide vaccines are not the whole pathogen, but rather only a fraction of part of them, for example, envelope of a virus or cell wall of a bacteria. This vaccine is safe and very efficient because the peptide used normally is the vital part of the pathogen, which make them useless when that part been attacked by immune system. Recombinant vaccine is also a fraction of the pathogen, but much smaller and less complex. One of the most common is the surface antigen of hepatitis B virus called HBsAg. The antigen is produced by cloning the antigen gene in yeast cells. You may refer to this table to see the common vaccines and their category. Highly virulent viruses, normally in form of inactivated or cellular fraction. And for additional input, you can refer to this diagram to see how different types of vaccines are being produced. This is a list of vaccine-preventable viral diseases and are widely used throughout the world. In future, we hope there will be more vaccines available against clinically significant viruses like dengue, adenovirus, and Coxsackie virus. In our country, these vaccinations are compulsory for every newborns, some only once, and some require few doses of booster. Now, we are going to look into vaccines which are used for some purposes other than newborn vaccination. This includes vaccination for adult females, special occupation, special lifestyles, environmental situation, special health status, travel, and against bioterrorism. For adult females, the second dose of measles, mumps, rubella (MMR) is given in adolescence. The purpose is to avoid the cases of congenital rubella among newborn babies. However, it shouldn't be given during pregnancy. For special occupations, depending on the job nature, the worker should get vaccines against virus or bacteria they commonly encountered. DT means diphtheria, tetanus, and type means typhoid A and B. BCG is the vaccine against tuberculosis bacteria. Special lifestyle vaccination program is common in countries where prostitution is legal or for inmates. The purpose is mainly to prevent sexually transmitted bacterial or viral diseases, for example, HIV and hepatitis B. Special health status person mainly refer to immunocompromised individuals. Since they are exposed to numerous infectious agents, they should get vaccinated against bacteria or virus, otherwise not compulsory for healthy individuals. However, the type of vaccine given to this group must be inactivated or dead vaccines for safety purpose. For travelers, they must follow the vaccination guideline of a country they are about to visit. Hajj pilgrims from India, for example, must receive cholera vaccine before leaving their country for Hajj. How safe are those vaccines? Nowadays, with the implementation of ISO in most commercial labs, people can rest assured that most vaccines are indeed safe, including the leaf attenuated version. Some of these points may be used by anti-vaccine groups, but these are minor when compared to the benefits of vaccines in preventing disease. We are going to look into antiviral drugs. As I previously mentioned, only few species of viruses are treatable by drugs. However, some of these antiviral drugs used only to ease the symptoms rather than killing the virus. Antiviral drugs are available for respiratory virus like influenza and RSV, hepatic virus like hepatitis B and C, herpes virus like HSV and varicella zoster virus, as well as HIV. As we know, influenza and RSV viruses require neuraminidase enzyme for exit from host cells, so the drugs include neuraminidase inhibitors, for example, oseltamivir and zanamivir. Another form of respiratory antiviral drugs prevent the virus from uncoating, so the virus cannot proceed with multiplication steps, for example, amantadine and rimantadine. The drugs to treat hepatitis B or C trigger the host cell, which is the liver cells, to produce enzyme which inhibit the viral RNA translation process, so the virus cannot multiply. These are examples of those hepatic antiviral drugs. Their mode of actions are different, but both aim to prevent the virus from multiplying by interrupting the virus central dogma process. Herpes virus infection is commonly treated using acyclovir or ganciclovir. Similar to hepatic viral drugs, these drugs also interrupt the central dogma process by inhibiting GTP cap addition to the mRNA and make it fragile. Ganciclovir is mainly used to treat cytomegalovirus infection due to its greater activity against that virus. Finally, antiviral drugs for HIV infection. The drugs do not kill the virus, but rather to suppress or minimize the viral replication, to restore immune functions, and to reduce morbidity as well as to improve the quality of life among the patients. If you have any question, feel free to text me or reply to this post. With that, thank you very much.